Friends, we are back with a new video. This time you can see that our household cooling system, that is the refrigerator, the food cooling system, we can just say in a very layman's word, the place where we keep the storage for foodstuffs, for storage for long-term preservation, and that thing is known as refrigerator. Now this refrigerator is a very good thing. We can preserve our foodstuffs and our many things fine, but it is a disturbing element also. It becomes a very disturbing element also. Why? It is because if we store these food grades, food elements for longer period, and if we do not clean the refrigerator properly, therefore what happens with the foodstuffs? This refrigerator wall of these refrigerators, actually the walls of the inner wall of the refrigerators get contaminated with the foodstuffs. Why? Because these foodstuffs from the sources we bring are not cleaned. They are not enough cleaned. So what they have the microbes or the microorganisms, and these microorganisms always gets cleansed, or they just cling to the internal wall of the refrigerators, and therefore they stay there for long periods. And if we do not clean them properly, they again come back to the foodstuff and they use the foodstuff as their food, and they again go back to and cling to the wall of the refrigerator. So this cycle goes on continuously, and there is a very heavy growth of the elements in the internal wall of the refrigerator. Our refrigerator has two parts. One, the general part where we keep the vegetables, other foodstuffs and things there, and the other one is the deep freezer, and there we keep the ice creams, we keep the milk, we keep the fish, animal matter, especially the animal matter is stored there for a long time in those refrigerators. So I have collected two samples from these two refrigerators, two compartments, one from the deep fridge and other from the general compartment. So here in this test tube you can see these two compartments. In this camera you can see these two test tubes. You can see one is containing the refrigerator main, one is named as refrigerator main. In the right side you will see this this is the right one. You can see the right one. This is the refrigerator. This is the right one. You can see from the right one. You can see that is the deep freeze. From this tube is from deep freeze, and this is from the other compartment, the main compartment. So this will be see this under microscope. So they are not these are just only the collections of washings. These are not haven't been put into culture media. So we are just going to see directly under the microscope. Then we will decide to put in the culture media. So just we will see this refrigerator content has settled down. So we have to use a vortex mixture just down here. It is not visible in the camera, but it's just down here. So we are going to put this in the vortex mixture. You will be able to hear the sound only. You will hear the sound. So just is vortexing. You can hear the sound. So it is done. So now the tube is done. So we are going to put the next tube. So it is done. So we are going to stop now the mixture. So this we will see under the microscope initially what it looks like. There is a mild detergent used in these washings. So very mild detergent. It is baking soda. So baking soda is mainly an alkaline medium. So it does not need to vortex a little bit. This main one. So here we are going to investigate under microscope these two separately under microscope what it reveals. So what kind of microorganisms we can see in the wall of the refrigerators, that we are going to see. Then we will decide to put it in the culture media or not, whatever. As you can see, this is the initial visualization of the refrigerator contaminants. This is the main part of the refrigerator, that is the bigger cabinet, the downwards cabinet, and you can see the molds. The crystallized particles are also seen. Now the microscope is focused and you can see the crystals. These crystals are of baking soda. So this should be eliminated, and you can see the black fringy molds. So the microscope is continuously getting focused. This is 200x magnification and you can see the molds. So more clear visions are getting done. So you can see here, you can see the molds. Also the molds, you can see this is initial stage of the molds. These are the flagellas, the molds with the flagellas. These are the floating molds with the flagellas. So black fungus may be there. We will be looking for it. Generally, black fungus stays in much more cooler region. We will investigate the deep freeze part. So these are the other parts. You can see now. You can see clearly the molds. These are the initial molds. Initial stage, molds in the initial stage. This is now the microscope is completely focused and you can see the beautiful molds are there, the flagellas are there, and Aspergillus may be there, black fungus may be there. So that we will see under, if we put this under culture, we will see it. So this is the mold, the initial investigation. So this is the second part of the investigation. Now this is the deep freeze part. You can see more contaminants here. As usual, the contaminants will be much higher, and the washings will be clearer because the dust accumulation inside the deep freeze is less. So you can see these are the crystals of this baking soda. So this should be eliminated, and you can see the flagellas. The same kind of molds are there. They are very similar. This mold formation is similar because contaminants goes from here to there, so the same contaminants are there. So anyway, let's see what anything bigger or anything fascinating we can see. These are the just baking soda particles. So this should be eliminated. This should not be taken in concern. We will, in the other part of the session, we will be removing this baking soda from this initial inoculant. We will be removing the baking soda and you can see the molds. Now you can see the molds clearly. So this is the other part. You can see an Aspergillus. This is the Aspergillus in the center of your screen. This is the Aspergillus. So Aspergillus is confirmed. So this is a busted Aspergillus. The head has busted. So anyhow, due to the scrubbing with the baking soda is a very good scrubber. It has busted. So anyhow, these are the baking soda particles, and you can see inside, you can see the molds, fringes and the molds. So Aspergillus will develop, definitely will develop, and you will be able to see this is the stealth. This is the contaminant stealth. So this is nothing just but contamination. You can just ignore it. And these are the initial investigations of these two samples. So this is a mold. This is also a very beautiful mold. Hello friends, we are back after 24 hours with our refrigerator cleaning solutions. That is our refrigerator cleaning extruder washing solutions. And as I have said in my previous video, that we are going to study this material, this thing. So in order to study this, we have to first put them in the culture media because it is fully contaminant. So there is nothing specific. We have to find out which is dominating, which fungus or which bacteria or which microorganism is dominating. That we have to find out. Is anything there dominating? Is there black fungus is present? Or Aspergillus, as I have showed you, the busted Aspergillus was there. One Aspergillus which has busted, its head has busted, disintegrated. So that was there. So maybe more will develop, or maybe more will be there. So in order to specify those things, we have to make a specific study. So for that we need to put it in the culture media. So before that there is a process. What process? The process of neutralizing this baking soda. If I neutralize this if we put this baking soda with the fungal base or the microbial base in the culture media, this baking soda will inhibit the process of culture growth. So therefore we have to eliminate this baking soda. So in order to eliminate this baking soda, we will be mixing this culture media, this sample, we mix this sample with glycine, which is an amino acid. Glycine is an amino acid. It is an acid. So therefore, it's a very weak acid, and it is a life-saving acid. It is a life acid. It is used in our life. It is used in our life form. That is the amino acid, required amino acid in our body. So glycine, if we use glycine, so there the bacteria will not die. If we use some other mineral acid, the process will be very fast, but the whole thing will be destroyed. So we have to keep those things. We have to destroy the baking soda only. But to neutralize the baking soda, we have to use glycine acid. So what the glycine will do, it will react with the baking soda and it will create a buffer salt, which not only will enhance the process of culture media or process of culture growth, rather it will idealize the molds. If there is a mold formation, it will disintegrate those molds. So it will idealize these molds. There will be a process of adialysis in this system. So it will react with the sodium baking soda, the sodium bicarbonate, or calcium bicarbonate, whatever it is. It will produces a calcium. Produces a calcium glycinate salt, which is a very good buffer. So therefore, we are using this glycine. We are intelligently using this glycine amino acid. So first of all, we will add some glycine solution to the buffer. It is around 0.2 molar glycine we will be using, and then we will centrifuge and remove the sodium glycinate or the calcium glycinate buffer, and we will just get the raw fungal matter or raw microbial matter from the refrigerants, and we will put them in the culture. And here we will be using RAMBACH agar, because RAMBACH agar is mainly used for contaminant microbial base. So when there is a contaminant base, everything is contaminated, mixed up. So from there the RAMBACH agar selects the actual dominating contaminant and it pulls it out, and it feeds and it grows, makes it to grow. So let us begin our experiment. Be with us in the later part of the video. Thank you. Hello friends, now we will be going to start the experiment. As you can see, this is our petri dish, and these are our centrifuge tubes. These two are our centrifuge tubes. There is a 10 ml centrifuge tubes. This is our glycine solution. These are the samples, and here is our RAMBACH agar. So first we are going to take a sample, this refrigerator washings, around 2 ml, into our first tube, in a 10 ml tube. So this is our sample. So a little bit is left out, so we will be taking all of that. So now this tube is empty. So here what we are going to do is we will be going to mix the glycine. So around 2 ml of glycine we will be using. 2 to 5 ml of glycine we will be using. So you have to take one tube, one pipette. This is our sterile pipette. So always use a sterile pipette. Don't use ordinary pipettes. That will contaminate your sample. So now we have to take the glycine, Just switching on the pipette and we will be taking the glycine. So this is our glycine. So it is quickly coming up. This is around 5 ml. So around this is 2.5 ml and we are using 5 ml glycine. So to initiate the reaction faster, so we are using around 5 ml glycine. Already the reaction has started. As you can see, in the camera that it has become smoky in appearance. It is not transparent, it is smoky in appearance. So the reaction has already started. So this will be eliminated by the smoky appearance is the calcium glycinate. So this calcium glycinate will be eliminated with the help of centrifugation, and then we will get the restia liquid we will be using as a sample. So this one is prepared, and now we are going to repeat with the second refrigerant washings. This is our refrigerant washings. So here we are going to take all amount. So this is little bit greater in amount. So anyway, no problem. We will just take this much only. So around 3 ml only. So now we will be putting this glycine. So here we will have to put 6 ml glycine, double the amount. So to ensure the reaction. So here we have to put so this is 5 ml and this is around 6 ml. Okay, now our solutions are ready. These are the two solutions. So now we have to mark. Now the question is we have to mark, because without marking we cannot proceed. So here is our two samples. So now we have to mark, and then we will go for proceeding to the centrifuge, and then we will start putting them into the culture media. So we have to take a little bit time from you. So I am taking a little bit time from you before going to the centrifuge, after going to the centrifuge, and little bit for culture, putting into the culture media. That I will show you later on. So friends, we are back after centrifugation, and as you can see, our two clear solutions are obtained, and here in this camera you will be able to see some precipitation is there. So that is the sodium glycinate or calcium glycinate, whatever, the baking soda, the salt of the baking soda and amino acid, glycine amino acid. So that is the precipitation. So this has been already been eliminated, and you can see the clear liquid over there. So this is looking clear, so that we will see. This is here also a little bit formation is there. You can see in the camera a little bit formation is there in the down. So these two are the baking soda eliminated with glycine amino acid. So now we will be using these clear liquids in our two flasks. As already there are two flasks are here. These are 250 ml flasks, and we will be using this liquid. So let us see what happens in this flask. So first of all we will inoculate this liquid. So first we are taking from the deep freeze, and okay, first let me name the flasks, because it will be after it will become confusing. So let us name the flask, and little bit cleaning is necessary, because before doing anything, whatever you are cleaning it or not, you have to put some alcohol and clean it. So here I am going to use some tissue paper and clean this flask. Because whatever you disinfect, after disinfection, whatever you have been done, before working, you have to clean it. Before working, you have to clean it. Otherwise, there may be some contaminants in between. So no contaminant should be encouraged at any stage of our experiment. So that we have to ensure first. With the help of a tweezer or a forceps, just grab the tissue paper inside and wrap it. This is alcohol, this is isopropyl alcohol, and just grab it. Grab the tissue paper with the forceps and take it out. This is an Alice forceps, so the gripping is very fast, very fine. So just tightly and take it out. With the flask, with the disinfectant, just take it out. So if there is any contaminant, this will come out. Okay, so it is there. Now the flask is absolutely clean, so there is nothing. So this you should do, and this one also. This one also. So this is apparently clean, so I'm not putting any tissue inside, so just it is clean. So now we are going to mark these flasks. This is very important. Always level your culture media, always level your flasks, always level your samples, everything. This is deep freeze, DF. And we have to deep freeze cleaning. We have to level it as 22, 23, 6, 22. So this is, and the name of the agar is RAMBACH agar. So this is one flask is ready, and another flask. So this is very important. This we have to do, and this is the refrigerator RF, and this is 23, 6, 22, and this is RAMBACH agar. So both are done. So now we will put our inoculate our specimens first. We will first put some culture media inside. So let us remove this Lichen to my table back, and we will bring this RAMBACH agar in front, and with the help of a pipette, we will take another pipette. So no problem. We will take another pipette. You have to keep this sterile pipette as many as possible in lab. Otherwise, you will be in trouble. So taking the culture media, so around 5 ml. So first we will put this in the deep freeze, and then another 5 ml. So 10 ml is required for each inoculant, because this is a big amount, so we have to take 10 ml, and another 10 ml for this culture media, for this flask. So this is coming out, and this will be delivered. Another, it is done. So it is over. So now we are going to put this inoculant. Inoculant should be taken accordingly as around 3.7 ml or rather 3.5 ml. Very slowly, not to disturb the. So this is RF. So I am putting it in the RF flask. So another. So inside the RF flask. So this is done. So this is done, and now from the defreeze. So from the defreeze, we will just remove this pipette tip, because maybe it may be different. It may be different, not the same, because the compartment is not the same. So now you are putting it in the different. So be careful and gone. So it is done. Now our work is over, main work is over. Now we are going to cork it. We need two corks, two separate corks. So I'm cutting two tissue papers and some cotton plug. As usual, I have shown you in my other tutorial videos that this is a self-made cork. These corks are disposable, so you have to make your own. So just folding the tissue paper like this and putting the cork. I am showing you in this camera. You can see I am putting the cork, and okay, I will show you again. So here is our tissue paper, folded tissue paper. Just this is our tissue paper, just fold it and hold it like this, then put the cork, put the cotton plug in the center and carefully roll it. So this is our cork done. So if you want to put some alcohol, spray some alcohol and just fold it like this. So make it tight, grip and put it inside the flask. So this is cork done. It's another tissue paper, fold it half the same way, just fold it half. This is a folded half and this is done. So you have to keep it here, take some cotton. We have to keep a cotton in the corner, and this much cotton will do. Put it inside and again same way roll it like this. So rolling it very nicely, and you can just make it a little bit thicker. Just press with your fingers and spray a little this alcohol and put inside the flask. Now these two are corked. So now they are ready for incubation for 5 days. 5 days or 10 days, whatever. According to the growth, you have to see the growth inside the flask. If the RAMBACH color changes the color, immediate start observation. If it does not change the color, keep for another 15 days. Or you can see any if you want to, if you have a very what should I say, the very questioning mind or very inquisitive mind, then you can study curiosity, out of curiosity, what is happening, then you can just see, open the incubator and take some sample around 3.5 ml and put it under tissue culture microscope and directly see what is there. Is there any growth or not? You can study that growth day by day. So that will be more interesting. You can compare the videos. You take the videos and you can compare the videos. So this was the growth, this percent of growth. Then you can see this is the percentage of growth. So you can make a curve that how the growth is happening day by day. So that is the research work. So that is one research work. So you can do it. So let us see what happens after 5 days. Hello friends, so we are back after 5 days with our refrigerator washings. As you can see in the camera, in our second camera, that they have changed the color. So they have become dark. So that means they are consisting of microorganisms and they are developed. So let us see. But as I have told you in earlier part of this video, that these microorganisms are contaminated, or rather this total culture media is contaminated. So they have different types of microorganisms. So they don't have any definite microorganism. So therefore we are not going to prolong this video with advanced studies and all that. So we will be ending this video with the second examination, and that will be our final examination. So this is just to show you, present you the presence of microorganism in the refrigerator. So after collecting the samples, keeping them in the culture, we can show you that we have shown you that this is just the presentation of presence of profuse microorganism in the refrigerator. So if the refrigerator is not cleaned properly, we will be having this type of microorganisms. So we are going to begin the experiment of just showing you the presence of microorganisms in the refrigerator. So now we are going to take around 1.8 ml, just 1.8 ml from the deep freeze. So we have here two different petri dishes. So we are just going to take the one petri dish and the other one in another petri dish. So if we change the tip and from by the second tip, we will be taking the second one and the second petri dish. So this we will just have a look under the SM400 microscope for our for visualization, to see what further microorganisms are developed and how much it has become. So that we are going to see, and after that we will be ending the video. So thank you very much for now. So you can see the refrigerator contents. This is the refrigerator from the big compartment, not the deep freeze. So you can see the lichens are there, the contaminants are there. This is basically a contaminant specimen. So different types of microorganisms are there. Mainly lichens are visible now. So these are the lichen molds actually. These are the black molds or the black lichens. This becomes the food of the black fungus. So eventually this will lead to the coming of black fungus. So these are the lichens. They form in the film in the layer, filmy layer on the inside compartments of the refrigerator. So they are also very dangerous. So they can contaminate the food and they can spoil the food also. Actually they help to rot the food inside the refrigerator, and these make the food vulnerable to the microorganisms. So watch it very carefully. So these are the lichens, the film lichens. So we are increasing the phase contrast and decreasing the light and increasing the phase contrast so that you can see the internal structure of the lichens. So this is a very thin film, less than a micrometer. You can see now, you can see the internal structure of the lichens. So these are very thin films. So these are the lichens actually. These becomes the food of the microorganisms. So mainly this specimen is contaminated. Nothing very important thing is visible. No specific funguses are there. So only these contaminant lichens are present. So this is we are going some other portion to see something more. So more lichens are there, more contaminants are there. So here we can see, so molds are there. So we are trying to focus, and tiny molds, and this is the patch. These are the patches. So these are also some mold patches, the dead mold patches. So these are the lichens, you can see. These are the lichens, and small contaminants, small molds are there. So this is also that mold. So only contaminants are found here. So no more such specific organism, I am telling again. So describing again, there is no specific contaminant. Some cryptosporidiums are also there. These are molds. These are scalular molds. So these are also scalular molds. Microscope is getting focused, and no such microorganisms are visible. So here we are ending the video of this part. So now we are in the deep freeze. Now you are watching the deep freeze contaminants. These are, you can see the cryptosporidiums are there. Lots of cryptosporidiums are there. Lots of molds are there. So floating molds, lichens are also there. So we are now here also we are searching for any specific fungus or any mold or something, but nothing is there. So only contaminants are visible. So these are the specific molds, and you can see a very big mold, big rusty lichen over there. This is a dusty lichen. It's a very thin film lichen. It's a very big one. We have to decrease the magnification to see the whole structure. So this is 200x magnification. We are you can see the internal structure of this lichen. It's a moldy structure, as you have seen in the beforehand. So these are the lichens, and we are decreasing the magnification to also decrease the light. We are now decreasing the light, and you can see this is now 100x magnification, and you can see the internal structure of the lichen, full structure of the lichen. So this is as usual like the before. So this is also a very contaminant specimen, and no such specific fungus found, but only contaminants are there. So this is also very dangerous. The contaminants are not safe, they are also dangerous. You can see different types of contaminants are there. They actually will bring the proper fungus. So if you leave the refrigerator contaminated for a long time, then also it's dangerous. So that is also bad. So anyhow, we will just put this, and we will just see other parts of the specimen. So only the lichens are visible, so nothing as such. So these are the floating contaminants. Some cryptosporidiums are there, but not such important. But it is contaminant. The pool of contaminants are there. So contamination specimen. This is the refrigerator. This refrigerator was under contamination, so it needs to be cleaned properly. So that also I will discuss in the later part of the video, that how to clean the refrigerator properly and disinfect the refrigerator time to time. So this is almost end of the video, and so anything else we can see. These are the patches, and almost end of the video. So this is a stealth, and the mold is there. Hello friends, we are ending the video now with our conclusion that, as you have seen in the microscopes, that contaminants are there, lots of contaminants are there, different types of contaminants are there, the lichens are there. So now the question comes that how should I clean the refrigerator? This is the most important part of this video. How to clean the refrigerator? Refrigerator can be cleaned in by usual household remedies. Just take a cup of distilled water or the drinking water, the water from the AGAR. You take it in a clean bucket or clean cup or clean saucer or clean pan, anything else. With a very clean cloth, dip the cloth in the baking soda, and just wipe the refrigerator inside the refrigerator after stopping the refrigerator. You have to shut down the refrigerator, and then when it comes into the normal temperature, then you clean it with the baking soda solution. And after that, after properly cleaning that, when the solution is clean 2 or 3 times, when you have to change the baking soda solution, you have to change it. After cleaning the baking soda solution, what you do, you take a cup of a flat cup. You should not take a cup, you should take a saucer or a flat cup, like that. The surface area of the cup should be more. It should be more like with a diameter of 8 inches or 9 inches diameter. Take that, or you can take a tray also, with a big tray, and fill that tray with vinegar. Just the household white vinegar. No, it's not an apple vinegar or any other vinegar, just a white vinegar, which is known as glacial acetic acid. Mainly it is known as acetic acid, 2% or 3% acetic acid. And keep that tray filled with vinegar one inside the deep freeze and another inside the main compartment, and keep the refrigerator shut down for about an hour. And then you remove those tray and run your refrigerator. So this will clean your refrigerator from all these type of lichens and your funguses and black molds and contaminations and all that. So thank you very much for keeping with us, and we are ending the video now. So thank you very much and have a nice day.